Good morning students. Our next topic is basic requirements of replication. In the before section I said about replication and the models of replication, and I also said about origin of replication. So I also give some of the introduction about the requirements of replication. So now I am going to say in detail about replication requirements. So first I want to say about the substrates. So which are the substrates used for the replication are 4-deoxyribonucleoside triphosphates. So here we say that 4 type of substrates are DATP, DGTP, DCTP and DTTP. So here DATP means deoxyadenosine triphosphate, and next one is deoxyguanosine triphosphate, deoxycytidine triphosphate and deoxytyrosine triphosphate. So these are the different types of the 4 types of deoxyribonucleoside triphosphates. So these are the substrates used in the replication process. So next one is templates. So here templates means if we having the parental DNA having double helical structure, 2 DNA strands. So these 2 DNA strands are acts as the templates and forms the daughter DNAs. So that are called as templates. We can use these templates for the synthesis of new daughter DNA strands. Okay, next ones are different types of enzymes which is used in the DNA replication. So first one is, so we have different types of enzymes and different types of proteins. So here DNA B protein, this is called as helicase. So it unwinds the DNA. And the next one is primase. So this is used for the synthesis of RNA primers for the extension of the strand. And the next one is DNA topoisomerase 1 and 2. So it will release or it can unwind the or it will cutting and joining single stranded DNA or both the strands. The next one is DNA polymerase. This can polymerize or elongate or it will produce the DNA. Okay. So that means it will produce the DNA chain elongation. And the next one is DNA ligase. So here ligase means attachment. It is used as a glue in this section. So it will attaches 2 strands of DNA. That is called as DNA ligase. Okay. The next ones are DNA A protein. So here we will use this protein is used for the duplex at the origin of replication. So we have the duplex, that means double stranded DNA, we call it as duplex. These double stranded DNA can be separated by DNA A protein. And the next one is single stranded binding protein. We called it as SSB protein. So here what happened is these single stranded binding protein will attaches to the origin of replication for the single strand and stabilize it, and it is used to degeneration by nucleases. It will help DNA from degeneration of DNA. Okay, next one is stir binding proteins. These are used in the termination process. So it prevents the helicase from the further unwinding and facilitates termination. So then we have to know clearly about the DNA polymerases. So DNA polymerases is used for the synthesis of DNA. Here it is works on the enzyme that is called as DNA dependent DNA polymerase. So here it is used for required for the DNA chain elongation, and it will repair 5' to 3' exonuclease activity. That means the starting line or starting strand is called 5'. So from 5' to 3', and it is used to act as the exonuclease activity. It is works in the outer side of the nucleus, and it will work at the activity at the outside of the nucleus, and it is also used for the proofreading. So here proofreading means during the replication we get different types of uh, problems. So to overcome these problems, these 3' to 5' exonuclease activity is useful for us. Okay, then we have different types of DNA polymerase. They are polymerase 1, polymerase 2, and polymerase 3. So here in eukaryotes we have alpha, beta, gamma, and epsilon also. Okay, so this is in prokaryotes we have 1, 2, 3, and in eukaryotes we have some of the subunits like alpha, beta, gamma, epsilon, and different types. So these have different types of functionings. So first polymerase 1 is used for the gap filling, and it is also used for the synthesis of Okazaki fragments of lagging strand, and then polymerase 2 is used for the proofreading, and it is also used for the DNA repair. Then polymerase 3, it is used for producing leading and lagging strands in the replication process, and some of the subunits like gamma and epsilon, it is used gamma and beta. Here beta is used for DNA repair and gamma is used for the mitochondrial DNA synthesis. These are the different types of polymerases. Then next, so we have to know in detail about the polymerase 1. So it is a DNA dependent DNA polymerase. It was first discovered by Arthur Korenberg in 1958. So for this production he received a Nobel Prize in Physiology and Medicine in 1959. So because he was discovered this, it is also called as Korenberg enzyme. Okay, next. So later DNA polymerase 2 and polymerase 3 were identified. These all having different types of biological activities. So these having 5' to 3' exonuclease activity and also polymerase activity. So polymerase activity means synthesizing the strands or attachment of the strands. So here polymerase means synthesis of DNA fragments. So here exonuclease activity means working these enzymes at the outside of the nucleus and became active in the outside of the nucleus. That is called as exonuclease activity. So here we have different types of organisms which have different types of DNA polymerases. We can separate different types of characteristics, and in eukaryotes and prokaryotes we have different types of polymerase activities of different types of subunits also. So we have in the structural genes we have polymerase A, polymerase B, and polymerase C, and we have different types of subunits. So in polymerase A we have only one subunit, and in the polymerase B we have 7 subunits, and the polymerase C having more than or equal to 10 subunits. So here we have different types of characters, and we have 3' to 5' exonuclease activity and 5' to 3' and exonuclease activity. It is present in both polymerase 1 and polymerase 3. So that means polymerase A and polymerase C. And these are the most important things and which is used in the replication process. So here 5' to 3' exonuclease activity is present in polymerase 1 and but it is absent in polymerase 2 and polymerase 3. So here polymerization rate also variation in different types of polymerase subunits. So here we have 16 to 20% in the polymerase 1 and 40% or 40 in the rate is in DNA polymerase 2. In DNA polymerase 3 we have 250 to 1000 polymerization rate. So we have different types of dissociation and association subunits here. That is about different types of polymerases, which is comparison with the E. coli and the normal organisms. So here what is the use of polymer exonucleases? So here 5' to 3' exonuclease activity can remove or excess mutated segments can be removed. Okay, so here primers or excess mutated segments can be removed by exonucleases activity. Okay, then here 3' to 5' exonuclease activity can excise mismatched nucleotides. That means it will add some of the nucleotides which are mismatched. So that means if there is any mismatch in that can be mismatched one will be separated and the other one is injected. That is called as 3' to 5' exonucleases activity. Here 5' to 3' means it will remove excess mutated or primers. Okay, so excess or mutated material will be separated by 5' to 3' exonuclease activity, and here 3' to 5' exonuclease activity is used for the excess mismatched nucleotides will be separated. That is the functions of the exonucleases. So here first one is DNA polymerase. So it is responsible for the proofreading and filling the gaps between the DNA during the damage or during the replication process. So here if we have a DNA, so it having N terminal and C terminal. So here if there is any cleavage, it is divided into two fragments. One is large fragment and the other one is small fragment. So here this small fragment having 5' to 3' exonuclease activity, and the large fragment having the Kellner fragment that having DNA polymerization. That means 3' to 5' exonuclease activity. Okay, then DNA polymerase 2. So here it is temporarily works when the DNA polymerase 1 and polymerase 3 are not in functional. Okay, so it is used when the absence of polymerase 1 and polymerase 2. So it is having the capability of synthesis of damaged templates, and it is participated in the DNA repair process also. And next one is DNA polymerase 3. So here a heterodimer enzymes composed of 10 different subunits are present. Actually it having the 10 different types of subunits and also having the high polymerization activity. That means 10 to the power of 5 nt per minute. So nucleotides per minute, and it can also use it for the different types of activities during the replication process, and it is used for the elongation process by synthesizing the DNA primers. So the next one is the structure of polymerase 3. It is the most important topic in this session, and this is one of the short question for the examination. So here we have different types of segments. The first one is alpha. It has it have 5' to 3' polymerization activity, and that means polymerization means that means synthesizing. So and we have the another subunit has 3' to 5' direction and having the exonuclease activity and plays an important role in the replication activity. Okay, so the next one is theta. Main, it is used for the heterodimer structure, and the preparation of the structure can be by the theta subunit. So these having different types of activities which are involved in the replication and also in the DNA mutations also, and it is helps in the DNA repair process also. And then RNA primase. So it is also called as DNA G. It is used for the synthesis of primers during the replication process by using free NTPs, and also it will use double standard DNAs as the substrate, as the templates, and primers is the short fragments, and it will be used of a several nucleotide ties along. So it is useful for the attachment production of the long slender DNA strand. So it is used by it, or it provides free 3' hydroxyl group to react the alpha particle atoms of DNTPs to form phosphodiester bonds. So the primates DNA B, DNA C, and an origin form a primosome complex at the initiation phase. So the initiation of the DNA replication can be started by the primosome complex formation. Next one is helicase. So it is referred to as DNAP. So helicase is attached to the one strand of the DNA, and it will help the DNA to overcome from the degeneration by nucleases. So it will opens the strands with consuming the energy in the form of ATP. The next one is SSB proteins. So we call it as single stranded binding proteins. These are used to attach to the DNA strand and make them to unwind, and it maintain the DNA template in the single strand form in order to prevent the double stranded DNA formation, and it also protects the vulnerable single stranded DNA from the nucleases activity. The next one is DNA gyrase. So here it will cut the DNA or reconnect the DNA strands of double stranded DNA and release the supercoil constants, and it also forms phosphodiester bonds. So it can change the double stranded DNA into the negative supercoiling state and consumption of ATP. So here we can call it as supercoiling 1 and DNA gyrase. We have two types, DNA gyrase 1, DNA gyrase 2. So here the next one is DNA ligase. So it is we called it as, in generally it is the glue which is attaches to DNA strands. So that is called as DNA ligase. In this it will use the phosphodiester bonds for the attachment of two molecules. Okay, so it connects to adjacent single stranded DNA strands by joining 3' hydroxyl group of one DNA and the 5' hydroxyl group of the other DNA, and seals the gaps between the different segments during the replication and recombination and also repairing processes. And the next one, the process of replication. So in how many stages DNA replication can be occur? There are mainly three stages of replication. The first one is physical separation of DNA strands. So physical separation means the daughter DNAs can be separated. So that is called, the parental strands will be separated. That is called as initiation. And the next one is elongation. So elongation means the separated DNA strands are converted into the daughter double stranded DNAs. That is called as elongation process. And the next one is termination. This is the process the replication will be stopped. That is called as, and the two pair daughter DNAs are formed, and stopping or completion of the replication process is called as termination. Okay, next one. So we have in this three stages. The first one is initiation. It is occurred at the origin of replication and separates the DNA strands. And the next one is elongation. It is the addition of primers and the formation of phosphodiester bonds and mismatch the correct bases and extending the DNA strands. And the next one, termination. So here stopping the DNA replication at a specific sites, that is called termination sites or ter sites. The first one is initiation. So the initiation is started at the by addition of primers. So it is started, actually the initiation is started at the OR site. Okay, so that is called as origin of replication. So here it is the binding of the primase takes place at the formation. It will form the primosomes which can recognize specific sites for the synthesis of the RNA primers. So here the replication fork will be formed. So during this, actually we can produce some of the bubbles. Okay, we called it as replication bubble. So the first DNA protein recognizes and binds to the ORI site or ORI of the DNA and successively denatures the DNA into two single strands. Okay, then helicase is then attached to the one strand of the DNA and forms a V-shaped structure for the synthesis. So here this region is called as replication fork. Then the stress is produced due to the unwinding of the helicase and released by the topoisomerase by cutting either one of the both double strands DNA. And the next one is the single stranded binding protein attaches to the two different strands which are separated by double stranded, which is separated double standards. So here the strands prevents their association by using the single stranded binding proteins. Then here it is called as a replication fork. So here we observe 3' and 5' and 5' to 3' and it we having the leading strands and lagging strands. Okay, so here one is continuous in strands are formed, that is called leading strands, and discontinuous strands called as Okazaki Okazaki fragments. These having discontinuous form of primers. So here the single stranded binding protein is attached to the DNA. So here helicase, SSP proteins, primers and DNA polymerase, these different types of substances are used in this initiation process. So here now the elongation. So during the elongation we will use DNA polymerase 3. I already said in the previous slides about the DNA polymerase 3. So it having the 5' to 3' direction. So exonuclease activity and endonuclease activity is also present. So during these it will form the primers. So both the DNA strands are synthesized simultaneously, but in opposite direction. One is in the direction towards the replication force and other one is outward direction away from the replication fork. So observe here, one is towards the replication and other one is outside of the replication fork. So here we produce leading strands and lagging strands. So here lagging strands means the strands which are produced in very small form and it having some of the gaps, that is called as lagging strands. Okay, the next one is leading strands. Here there is complete synthesis of strand which do not having any gaps, that is called as leading strands, and the gaps having the sequences, that is called as Okazaki fragment. Here the RNA polymerase the RNA primers are attached and primers are forming one by one. Okay, so here observe this picture. These picture having the helicase, topoisomerase, SSP proteins and also RNA primers. Okay, so direction of movement of the replication formula. This is the process of DNA replication. This is the elongation process. The primers are attaching to the DNA. That is called the doubling. That means elongation process. Here, the first parent strands are separated. Then, from each strand, we can produce different types of primers. So, one having leading strand, other one have lagging strands. So green color strands are parental strands and red color strands are daughter strands. These daughter strands are produced during the elongation process. Okay, here I also said about lagging strands and leading strands. So lagging strand means having some of the gaps between the strands. It is also called as Okazaki fragments. Okay, then leading strands are having without gaps, it will be produced. Okay, so here Okazaki fragments. So these are between 1000 to 2000 nucleotides long in a prokaryotes and approximately 150 nucleotides along the eukaryotes. So here at the 10th nucleotide RNA polymerase, they are separated. So two Okazaki fragments are connected into continuous newly synthesized complementary strands by using the enzyme ligase. So here two different DNA strands are attached by the ligase. So ligase is the enzyme which is used for the attachment of two DNA molecules. Okay, so here this is the ligase. So this ligase is used for the attachment of the two strands, one daughter strand and the other one is parental strand. During this the gaps, the discontinuous form of primers can be attached by this ligase enzyme. So here next one is termination. So we get two from one DNA strand, we can get two daughter strands. That is in the elongation process. The elongation process is completed. The next one is termination. Here the termination sequence is ter. So here direction of termination of replication will be started. So a specific protein, ter binding protein, will bind to the sequence and prevents the helicase from the further unwinding of the DNA, and almost all proteins which are used in initiation elongation are separated in this section. So a helicase will be moved, primers, RNA polymerases will be moved and facilitates the termination of the replication. This is the process of the replication. Okay, the next one is proofreading. Here proofreading means the error ratio during the replication is kept very low. Here the what are the errors during the replication will be very low. That is called as proofreading. So here we have during the replication we have many errors. The decreasing the number of errors is called as proofreading. So proofreading is most important to know the accuracy of the DNA, how it will produce or synthesize different types of daughter fragments by using 3' to 5' exonuclease activity. So here DNA polymerase and 1 and 2 are known to excise or remove mismatched nucleotides before the introduction of next nucleotide. So this is called as proofreading. So the next topic is inhibitors of replication. So here replication can be inhibited by some of the substances like different types of antibiotics like novobiocin, nalidixic acid and also ciprofloxacin. These three are the antibiotics which can inhibit the DNA replication. So it is only mainly done in the prokaryotes, and in eukaryotes, that means in humans, it is widely used as the anti-cancerous drugs that are adrimycin, camptothecin and ectopsid and doxorubicin. These different types of antibiotics are used as anti-cancerous drugs. During, if we use this continuously, it will stop the inhibition of the DNA replication, and these different types can work on different organ. When we will use these types of antibacterial drugs, the inhibition of replication will be done due to using these drugs like novobiacin, nalidixic acid and ciprofloxacin. So if we use these drugs in the prokaryotic organisms like bacteria, the replication will be stopped. Okay, so here during this, these topoisomerase inhibit are widely used to treatment of urinary tract and other infections. If we use some of the drugs for some of the health to remove or recovery from the illness, during we can give uh, um, some of the diseases or some of the inflammations to the organisms. That one is replication inhibition is one of the certain type of process that can be given by some of the antibiotics. So using some of the antibiotics increment or decrement, that means avoiding some of the antibiotics is best result for the human beings from recovering of some of the diseases. This is one of the major one which if we inhibit this replication, it is mostly harmful to the organisms. If we have any pains or some of the problems for health, if we use this type of drugs, we can eliminate or elucidate some of extra diseases by these antibiotics. So these antibiotics are some of the drugs avoiding is the best process for the organisms. So certain types of anti-cancers and antiviral drugs inhibited elongation of DNA chain by incorporating certain nucleotide analogs. So this is also most important one to understand about the inhibition of the DNA replication. The next one is uses of DNA replication mutants in the study of replication. So some of the environmental mutagenesis can causes the inhibition of some of the replications, and it will also disturb the replication by damaging the DNA and leading to the cancer to the other diseases. And these DNA damages are levels of DNA replication by a single polymerase to the complex DNA replisomes, and some of the diseases can be attached to the organism by the mutants which are going to mutations in the organisms. So the DNA damage can affect the replication in three major ways. First one is reducing replication fidelity. That means fidelity means the frequency will be reduced. And causing frame shift mutations. Frame shift mutations means we will study in the next unit. So here frame shift mutations means some of the, if we have, for example, if we have some of the frame or the sequence, so in that sequence if you have A B C D E is the sequence of that that amino acid or that character. If A B C D E is cutted at the middle of the sequence, if A B is present and C is removed, D is present, that is called, that means the removal of some of the sequence some of the amino acid in between the sequence, that is called as frame shift mutation. And due to this frame shift mutation also, the characteristics of an organism will be changed, and it is also blocking some of the replication process. So these three are the different types of replication process will be damaged by these different types of mutagenesis. Okay, then so these are the major important to know about DNA mutants during the DNA replication. And these are the most one, and this can bind that DNA mutants will attach it to the DNA and stop some of the sequences, and most DNA polymerases catalyzes each single stranded DNA base and incorporation cycles according to the general mechanism, and which will binds to form binary complexes and selectively binds to correct DNT piece based on Watson-Crick model of DNA or Watson-Crick model of base pairing from polymerase DNA tipis. So, and forms the ternary complex. Then the inducing a conformational change to facilitate the formation of phosphodiester bonds. So, these are the major calamities or major sequences or major functions which are done by the mutants, and it will damage the DNA replication, and it also inhibits the DNA replication. So to overcome these, we can avoid to using some of the chemicals, some of the drugs, and some of the antibiotics. Okay, thank you.